morning, children. In the last class, we were discussing about the transcription in prokaryotes and eukaryotes, right? Yeah. So after that one, let us study today the very important topic, that is the genetic code. Okay. So let us study about the genetic code in this class. Fine. Yeah. First of all, question arises: how can you define genetic code? Okay. Yeah. Let me to explain. So it can be defined as, that means the sequence of nucleotides or nitrogen bases in messenger RNA, so that contains the information for protein synthesis. It is commonly used as a translation. Okay, so what is the genetic code means? It is a sequence of nucleotides. They are commonly used as nitrogen bases in messenger RNA, so that contains the information for protein synthesis. So it is also known as the translation. Okay, so in other words, the coded message or genetical information present on messenger RNA strand is decoded into a polypeptide chain. Okay, so that is nothing but the protein. It is also called genetic code. Are you getting how can I define the genetic code? So you can write the definition, that is a sequence of nucleotides in messenger RNA that contains the information for protein synthesis, or in other words, the coded message or genetical information present on messenger RNA strand is decoded into a polypeptide chain is called genetic code. Okay, so generally how it will be? See, the sequence of 3 bases determining a single amino acid is called codon. Okay, so here only the sequence is given. So for example, in this sequence, beginning from 5 prime to 3 prime direction, there are about 9 codons are present. The first codon AUG, second one UAC, third one is GUA, fourth one is CGU, fifth one is ACG, sixth one UAC, seventh one GUA, eighth one CGU, ninth one UAA. So this is nothing but the sequence of the 3 bases, that is 3 nitrogen bases determining a single amino acid. It is called codon. Okay, so these codons are responsible for the synthesis of particular protein. Okay, so these codons are present on messenger RNA, so they are responsible for the synthesis of particular protein. Okay, yeah. So after this one, when it comes to the discovery with respect to the genetic code, there is a name of the scientist, George Gamow. He is the first person who suggested that for coding 20 amino acids, the code should be made up of 3 nucleotides. So thus there are 64 codons are present. Okay, according to, so that is a George Gamow only. So that means total number of codons, it will be is equal to 4 to the power of 3. That means 4 into 4 into 4. 4 4s are, that is 16, and in 16 4s are 64. So this is how actually George Gamow suggested that for the coding of the 20 amino acid. Fortunately, unfortunately, when they tried to check out with respect to the genetic code, it is come to know it is true. And after that one, there is a very important person. You must have heard about this name of the scientist, Hargobind Khurana. Okay, so actually he is also got the Nobel Prize for the discovery of these genetic codon sequences. Okay, even he is actually Indian. Okay, Indian. So but later on he moved towards the USA. Okay, yeah. So Hargobind Khurana developed the chemical method in synthesizing RNA molecules with a defined combination of the bases. So they are nothing but in the form of homopolymers and copolymers. For the purpose of his discovery, finally he got the Nobel Prize. Okay, yeah, so he shared the Nobel Prize with respect to George Gamow. Fine. And so then after, there is another important person who comes under the discoveries with respect to genetic code, the Marshall Nirenberg. Marshall Nirenberg, one who developed the self-read system for the protein synthesis. Okay, are you getting what I'm telling? Yeah. And there is another name of the scientist, or the generally one who studied about the genetic code or molecular biology. Okay, Severo Okawa enzyme. It is popularly known as, that is, Severo Okawa enzyme polynucleotide phosphorylase. Okay, is used to polymerize the RNA with defined sequences in a template independent manner. So these are all the very important discovery with respect to the genetic code. Okay, and these are all when they try to check out by using the electron microscopes, it is come to know the truth also, and whatever they proposed absolutely correct. Okay, present also, so we are using the same things. Fine. Yeah. So after this one, when it comes to the what are the amino acids, okay, which is generally coded by these genetic codes. See, whatever the combination of the genetic codes, okay, as I said, now only that is 4 to the power of 3, 64 codons are there, isn't it? Yeah. So it means out of this, that is the 64 codons are existed. So these 64 codons except the 3, so that means UAA, UAG and UGA, so they are called terminator codons, which do not code for any amino acids. Hence the remaining, okay, 61 codons, they code for 20 amino acids, which can be identified in case of the whatever the unicellular and multicellular animals. Okay, so what are those 20 types of the amino acids involved in the translation in prokaryotes as well as the eukaryotes means? So let us discuss what are those one. See, these are all the amino acids which is important for the purpose of competitive. Okay, yeah. So consider here on the right side they have given the combination of the different amino acids, but whatever it may be, they're all the combination of 20 amino acids. Okay, yeah. For example, first one is alanine. Okay, it can be written as a ALA. Arginine, short form ARG. Asparagine, ASN. Aspartic acid, cysteine. Glutamine, glutamic acid. Glycine, histidine. Isoleucine, leucine. Lysine, methanoine. Phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine. So these are all the 20 types of the different amino acids involved in the translation, which is coded by the 61 codons. Okay, except those 3. So that is nothing but the terminator codons. Okay, yeah. See, here is the table which is indicates or containing the codons for various amino acids. So they are also known as the genetic code dictionary. What it's containing? See here, so there are the 3 combinations, the letters with respect to the tables. Okay, so consider, okay, so in the beginning only, if you observe how they are responsible for the synthesis of particular codons. In the first one, okay, if you see that UUU, UUC, which codes for amino acid phenylalanine. Okay, at the same time UUA, UUG, okay, and even CUU, CUC, CUA, CUG. So these are all the codons which code for amino acid leucine. Okay, and we can also identify there is another one, GUU, GUC, GUA, and GUG. So these codons codes for amino acid valine. Okay, so like that, so we can identify the genetic codes. Okay, the genetic codes for the various amino acids, so that this combination of these 61 amino codons, they are responsible for synthesis of 20 amino acids. Okay, yeah, so this table is also very important, and they can ask the number of times already. They were asked in the competitive also. Okay, so with respect to the particular codons as well as the synthesis of amino acids, like that they will give for match the following also. Okay, it is better to write and practice, but in theory they may not ask, but generally in competitive they will ask. Okay, which is related to the genetic code dictionary. Fine, yeah. So after this one, so we have to discuss about the salient features of genetic code, or it can also be called the features of genetic code. Okay, yeah. So what are the features of this genetic code? So in generally this question they can ask for minimum 3 marks questions, or sometimes they can also ask for the 5 marks question also. Now let us discuss one by one. What are the features of genetic code, or the salient features of genetic code? And that one, first one is, that is triplet codon, or we can also say the codon is triplet. What is the meaning of this? See, generally whatever you observe the codons, the genetic code is a triplet, isn't it? So that is, the codon is represented by 3 nucleotide sequences present on messenger RNA, isn't it? The messenger RNA strand that codes for a type of amino acid, isn't it? Yeah. And after this one, the 61 codes for, that is the 61 codons codes for amino acids. Do you observe? Yeah, as I said in the beginning also, see, so in triplet codons, 64 codons are existed. And totally how many number of codons are existed? 64. But out of these 64, 61 the 61 codons code for amino acids. The remaining 3 codons are called, yes, they are nothing but terminator codons. Why? So because UAA, UAG and UGA do not code for any amino acids, so they are acts as a stop codons. They are also known as terminatory codons or nonsense codons. Okay, yeah. And because of these few amino acids which is code for more than 2 codons also, so sometimes this phenomena is called degeneracy or prudency. Okay, and there is another one, see, a very important one, AUG. The AUG codon has dual function. Okay, AUG codon has a dual function. So that means the AUG, it codes for methanoin, also acts as a initiator codon. Okay, so that means the process of translation generally initiated by the initiator codon AUG that codes for the amino acid methanoin, isn't it? At the in case of the AUG is absent, GUG also acts as the initiator codon. But so here the methanoin is the first amino acid and the formal methanoin in case of the prokaryotes. Okay, so as I said in the beginning also, an amino acid is coded by many codons except AUG for methanoin and UGG for tryptophan. Such codons are called degenerate codons. Okay, such codons are called degenerate. Okay, so they can also call, what is the meaning of the degeneracy or redundancy? Okay, so then you should write In a triplet codon system, 64 codons are existed, in which 3 codons such as UAA, UAG and UGA are the terminator codons, which do not code for any amino acids. Hence the remaining 61 codons for 20 amino acids, because few amino acids are coded by more than 2 codons. So this phenomena is called degeneracy or redundancy. Okay, so that's why they're also known as codons are degenerate codons. Can you identify here? Yes, UAA. Okay, so or else we can also identify UAG or UGA. Are you able to identify? Yes. And the next one is, generally while we are telling about the characteristics or the salient features of the genetic code, we can also say the codons are commaless, or it can also be called no punctuations. Okay, no punctuations between the adjacent codons. Okay, so the codon is read in messenger RNA in a contagious fashion. What is the meaning of this one? See, if we observe the properly, the codons present on messenger RNA strand are translated or decoded continuously without stop unless it reaches the terminator codon. Okay, so that's why, so this feature is also called codons are commaless, or we can also say no punctuations or no punctuations. Fine. And the next one is very important one. The genetic code is universal. What is the meaning of this one? You know, the genetic code is common and found in all the living organisms. If I take an example from the bacteria to human, UUU is a codon that's code for amino acid phenylalanine in all the organisms. Okay, so only some exceptions are found in mitochondrial codons and in some protozoans. Except that, exceptional cases, remaining all. So that means remaining all. So they show the characteristic feature, that is the genetic code is universal. Okay, I hope you can identify easily. Fine. Yeah. Exception is only in mitochondrial codons and in some protozoans, but it is a rare cases. Yes. The next one is genetic code is non-overlapping. Okay, so genetic code is non-overlapping. What is the meaning of that one? That is non-overlapping. See, if we observe whatever the codons which is present on the messenger RNA, each 3 successive nucleotides present on messenger RNA strand is a codon that's code for a specific amino acid. Hence, a letter or nucleotide of one codon never shares with another codon. Okay, so that's why, so this characteristic is also known as non-overlapping. Okay, non-overlapping. Then the next one, the feature or the the salient features of the genetic code. The genetic code is unambiguous and specific. Okay, it can also be called the sensible also. A specific codon codes for only specific type of amino acid during protein synthesis. So that means one codon specifies only one amino acid. Okay, so if you take example, AUG is a codon that codes for amino acid only methanoin. Okay, so this is what, so these are all the considered as the features of genetic code. Are you able to get it? Yeah. So and here when it comes to the next topic with respect to the genetic code, okay, so very important topics. So as earlier we were also discussed, but it is related to the genetic code. You must have heard about the mutation and the genetic code. I told you how can I define the mutation. It is sudden heritable changes happens in the codon sequences, isn't it? And the genetic code, how it will be. See, the relationship between DNA and genes are best understood by mutation studies. Okay, because you know very well that in the last chapter, the last topic, while we are discussing about the genetical disorder, you must remember. Okay, so the deletions and the rearrangements in a DNA may cause due to the loss or gain of gene and so a function. So that means, so there are two possibilities we were discussed, isn't it? In that one, first one is the point mutation. Okay, did you remember that? What is a point mutation? Yeah, the point mutation, it occurs due to replacement the nitrogen base within the gene. Okay, and also it only affects the change of particular amino acids. In best understood the cause of sickle cell anemia, or we can also call it the best example for point mutation is sickle cell anemia, isn't it? And there is another type of the disorder we were discussed in the earlier also, frame shift mutation. Did you remember about that one? Yeah, how it will happen? The frame shift mutation occurs due to insertion or deletion of one or more nitrogen bases in the gene. Okay, yeah. So there is a change in whole sequence of amino acids from the point of insertion or deletion. The best understood in case of beta thalassemia, isn't it? So that's what, as I said, the mutation and genetic code there. So that means the relationship between DNA and the genes are the best understood by mutation. Okay, so that's what I said. The insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion. It is also called the frame shift insertion or deletion mutation. Okay, so here only you can identify whenever the insertion is happening, there is addition of an extra, that is a base pair G. Okay, then the whole reading of the frame shift is changed. Are you able to get it? The middle one, it is actually a normal sequence. Fine. And here, in case of the deletion, in the lower one, if you identify that one, what is happening? If the A is missing, the complete reading frame is get changed. So this type of the mutations can be studied. Okay, this type of the mutations can be studied with respect to the relationship between DNA and the genes, so that we can understand what is the reason for exactly the point mutation as well as the frame shift mutation. Okay, yeah. And after this one, see, as I said, here only when it comes to the insertion or deletion of 3 or its multiple bases. In the last one, we were discussed about only point 1. Here, if the insertion or deletion of 3 or its multiple bases insert or delete one or multiple codes, the reading complete frame remains unaltered from the point onwards. Hence, one or multiple amino acids are inserted or deleted. Okay, so that it proves that the codon is triplet and is read contiguously. Okay, so it reads contiguously. Okay, yeah, so this is what, so it can be studied with the help of frame shift mutations. I hope you can identify the normal sequence, the differences between insertion, the GAG complete, so bases get added. What about in case of the deletion? Complete ACG, that is bases get deleted. So this type of the mutation is also called frame shift mutation. Okay, yeah. So after this one, we have to discuss about the types of the RNA. So this is very important. Yeah, I told in the beginning also. So RNA not only acts as a genetic material, isn't it? It also acts as a catalyst also. Okay, so there are three type of RNA we can identify. What are those three type of the RNA? Yes, messenger RNA, ribosomal RNA, and transfer RNA. Let us discuss one by one the types of RNA. Okay, so in that one, first one is, yes, messenger RNA. So the word itself indicates which provides. So that means messenger RNA which provides template for translation. Okay, it is very important for the synthesis of particular protein. Okay, so then the second one is ribosomal RNA. So in the short form it is called rRNA. It is very important. The ribosomal RNA is acts as a structural and catalytic role during translation. The very best example, the 23S ribosomal RNA in bacteria acts as ribozyme. Okay, because this ribozyme perform the structural as well as the catalytic role during translation. Okay, so and the next one is tRNA. So there is a transfer RNA. It is also known as sRNA. Okay, why it is called sRNA? The soluble RNA. It is the only type of RNA it can be soluble. Okay, so that means tRNA brings amino acids for protein synthesis and takes the genetic code. Okay, I hope you can identify exactly the types of RNA. Okay, the messenger RNA which is present in the nitrogen bases, and the ribosomal RNA, and later, yes, tRNA. Fine, yes. So here is one of the best example which is explained about the Francis Crick postulated the presence of an adapter molecule can read the code and to link with the amino acid. You must have heard about generally. That means the question, whenever the question arises, why tRNA is referred to as an adapter molecule? The answer is clear, since tRNA carries an amino acid that attached to that is coded by codon present on messenger RNA. Okay, I will repeat once again. Why the tRNA is referred to as an adapter molecule means you should write the answer. Since tRNA carries an amino acid that attached to, so that is coded by an codon present on messenger RNA. Okay, and remember, so because it has an anticodon NODOC loop, so that has the basis complemented to the codon, and even an amino acid receptor end to which amino acid binds, a ribosome binding loop, enzyme binding loop. Okay, so this can be best explained by, so that the structure of the tRNA can be best explained by using cloverleaf model. Okay, so that's what I just now said. So the tRNA, okay, so when we start the discussion with respect to the RNA, that molecule transfer amino acid to the site of protein synthesis. Okay, and into the ribosome during protein synthesis, hence the name is called adapter. and the tRNA is called soluble RNA. So but remember, when it comes to the percentage, the tRNA, it makes about 10 to 20% of the total cellular RNA, and it is the lowest among the RNA among the three types of the RNA. Are you getting what I'm telling? Yeah. So let us discuss the cloverleaf model of the tRNA. Yes. See, actually, the cloverleaf model of the tRNA was proposed by Robert Holly. Okay, please note down. The cloverleaf model of tRNA was proposed by Robert Holly. R-O-B-E-R-T, Robert Holly, H-O-L-L-E-Y. So this resembles the leaf of clover plant. Actually, why they have given only this name only? The structure of tRNA, which resembles the leaf of clover plant. The botanical name of the clover plant, Trifolium dubinum. Okay, and the tRNA is made up of single polynucleotide chain consisting of CCA nucleotide at 3' end. Okay, and there is an amino acid binding site to which amino acids binds during translation. Okay, are you getting what I'm telling? Yeah. And the another region is ribosomal recognition type. That is a site. It is also called T summation C loop that recognizes the ribosomes during protein synthesis. Okay, yeah. So for the initiation, there is another tRNA is called initiator tRNA. So there are no tRNAs for stop codons. Remember, the secondary, the 2D structure of tRNA looks like a clover leaf, but 3D structure of the tRNA looks like inverted L shape. Okay, so there is a difference between 2D and 3D structures. Okay, yeah. And when it comes to the exact structure, as I said, there is an anti-codon site consisting for 3 nucleotide sequences which are complemented to the respective codons present on messenger RNA strand. Hence, they are also called NODOC. N-O-D-O-C. NODOC structure. Fine. Yeah. And there is a DHU loop. I hope you can identify DHU loop. So this DHU loop is also called dihydroxyuridine loop, the containing amino acid active enzyme called amino acyl synthetase. Amino acetase, that is, it is also called amino acid acceptor end. Fine. And each tRNA binds with specific amino acid. Hence, as there are 61 amino acids, there are at least 61 types of tRNA found. Okay, and finally, one specific tRNA with anticodon UAC called initiator tRNA. Initiator tRNA, as it carry an amino acid methanoin coded by initiator codon called AUG. Okay, and there is no tRNA for stop codons. So that is called UAA, UAG and UGA. Okay, so you should be careful and to identify the structure exactly. Let me to tell you once again briefly with respect to the interesting factors about the cloverleaf model of the tRNA. So it was proposed by Robert Holly et al. In 1965. According to this model, a single polynucleotide chain of tRNA coiled to form a cloverleaf-like structure. Okay, so it is consisting of the following feature: an acceptor arm. An acceptor arm, it has 3' end with CCA as the terminal nucleotide to this particular amino acids binds, and DHU loop, dihydroxyuridine nucleotide, it has amino acid activating enzyme recognition site. And another one is T summation C loop, it has the ribosome recognition site. And anticodon loop, anticodon loop, it is a codon recognition site. It contains a sequence of 3 nucleotides called anticodon. Okay, so that's why it is also called codon, capital letters CODON, NODOC, that is a anticodon. And with variable arm, it consists of few nucleotides. Are you able to identify the structure? And for initiation, there is specific tRNA known as initiator tRNA having anticodon UAC, but there are no tRNAs for stop codons. Okay, so this is with respect to the tRNA, the structure of the, so that is the adapter molecule. Okay, so today what we will discuss? Today we will discuss about in detail about the genetic code, the features or the properties or general characteristics of the genetic code. Then after we will studied about the types of the RNA and also the tRNA as a adapter molecule and the structure of tRNA as a adapter molecules. Okay, so let it be. Next class we will continue the remaining topics. Thank you. Have a nice day.